Okay, a happy day for my students and my viewers. And today we are going to discuss about the cell and its structures, because the first unit of this first portion, that is the very important topic of this biomedical instrumentation, because without knowing this cell and its structures, it is waste of studying all the topics, because this is the basic one. Okay, so this is Narajan, working as an assistant professor in the Department of Electrical and Electronics Engineering, School of Engineering, Trichy, aggregated by NAC, approved by ICT New Delhi, applied to Anna University Chennai, and let us go to the topic. Yes, now cell and its structures. Okay, so our human cells and animal cells resemble the same. Okay, when we view through the microscope. Okay, so here there are major compositions of the cells. Very important thing is cell membrane. Okay, this is the outer cover of the cells that protects the inner parts of the cells and gives the shape to the cells. And next one is rough endoplasmic reticulum. Okay, rough endoplasmic reticulum, RER. Next one is lysosomes. Okay, next cytoplasm, that is an important one. Then vacuole. Okay, vacuole is there. Then centrosome. Okay. Next one is mitochondria, a very important one. That is also called as the energy storage units. Okay, then nuclear membrane. Okay, nuclear membrane, it is a double layered. By looking at the image itself, we can understand that that is a double layered which protects the inner nucleolus. Okay, from the cytoplasm and as well as from the other agents. Then finally, nucleolus is the center portion of the cell, and nuclear pores is there. Microtubes, okay, microtubule. Then SER, that is smooth endoplasmic reticulum. Smooth endoplasmic reticulum. Finally, Golgi complex. Okay, it is the energy producing sections, and ribosomes. Okay, ribosome is nothing but it is a sac that synthesizes the protein, and granular endoplasmic reticulum. Okay, granular endoplasmic reticulum. So from this, you can able to understand that endoplasmic reticulum is, there are two types. Okay, actually it is in three types. First one is rough one, next one is smooth one, and finally granular. Okay, going to the next portion. First one is plasma membrane or cell membrane. Okay, plasma membrane or cell membrane, it is composed of double layer of phospholipids, in which proteins are embedded. Okay, so it gives this, as I said earlier, it gives the shapes to the cell and allows the passage of the materials into and outside of the cells. Here, as far as in this subject, it is concerned about the transfer of ions, sodium ion, potassium ion, chloride ion, everything. Okay, this is because we are discussing about the biomedical instrumentation and the biopotentials. Next one is cytoplasm. Cytoplasm, it is a fluid, jelly-like substance between the cell membrane and nucleolus. Okay, between the cell membrane and nucleolus. Okay, so if you look at this cell membrane and nucleolus, the entire whole thing, okay, the entire whole thing is composed of cytoplasm. In this cytoplasm, these components are embedded. Okay, thus lysosomes and vacuoles, centrosomes, mitochondria, everything is embedded in the cytoplasm. Okay, so it serves as a medium in which chemical reaction occurs. That's all. Okay, it is a medium. It's where the chemical reaction occurs. Then endoplasmic reticulum. Okay, the system of interconnected membrane forming the channels and tubes. Okay, that endoplasmic reticulum, already I said that there are three portions in this endoplasmic. Rough one is there, smooth one is there, and adnular is there. Okay, so again here it is classified into, majorly classified into two types. One is smooth one and another one is rough one. Okay, smooth one is adnular and rough one is granular. Okay, so adnular type metabolizes the nonpolar compounds. Okay, nonpolar compounds and stores in straightened muscle cells. Okay, then granular type assist in protein synthesis. Next one is ribosomes. Okay, ribosomes is nothing but ribonucleic acid. Okay, it is nothing but RNA. It is ribonucleic acid. It synthesizes the protein. Golgi complex. Golgi complex. It is a cluster of flattened membrane sac. Here already we discussed, we come across this word flattened membrane. Where we come across this word flattened membrane? Here. Okay, flattened membrane is straightened muscles. Flattened membrane is like straightened muscles. Okay, so what is its role? It synthesizes the carbohydrates and packages muscles for secretion. Okay, it secretes lipids and glycoproteins. It separates lipids and glycoproteins. Next one is mitochondria. Okay, it is again hereafter whatever the components that are present in the cytoplasm will be in form of membranous sac. Example, you see this mitochondria, it is a membranous sac. Then lysosome, it is a membranous sac. Then peroxisome, it is a membranous vessels. Then centrosome, it is a non-membranous except the centrosome. Okay, then vacuole, it is a membranous sac. Again, this microfilament and microtubes, it is also a membranous sac. Okay, it is of thin and hollow tube. Tube and sacs, it is a straightened one. It is it does not have any definite shapes. That's all. So all these were the membranous sacs. Okay, let us come into this individual. Mitochondria, it releases energy from food molecules and transfers into that is active particles. Okay, active particles. Next one is lysosomes, digest foreign materials, that is molecules, and wounds and damaged organisms. What is the role of this lysosomes? It is a digestive. Simply we can compare this one with WBC. What is the role of WBC? WBC, W is white blood corpuscles. It invades the foreign materials and digest them. Similarly, it is also the same. Okay. Then peroxisomes, it contains enzymes that detoxify harmful molecules and break down hydrogen peroxide. Finally, centrosomes. Centrosomes helps in organizing spindle fibers and distribute chromosomes during the mitosis. Mitosis, the cell division, at the time of cell division, because the centrosomes will evenly divide the cells. If there is any deficiency in during the that is cell division, then it creates the we can say that one as a malfunction, and in terms of engineering it will be called as malfunction, that is some indeficiency. Okay, sorry, it is a deficient one. It is not evenly balanced. We can compare that one like this. And vacuoles, it stores and releases various substances into the cytosomes. Okay, and finally microfilament and microtubes. Okay, again it supports the cytoplasm and transport materials within the cytoplasm. And finally cilia and flagella, and it is an projections extended from the surface of the cells. Okay, and Now this we can compare this one corona. Okay, corona will be having a very spike-like structures, but actually this is not a spike-like structures. It is just like the projections. But this is not that much very big and huge. Okay, it is just like simple one. Okay, and moves particles along the cell surface and moves the cells. And the next one is nuclear envelope. This is also a double layered membrane. Okay, it is composed of proteins and lipid molecules. And what is the role? It supports the nucleus and controls the passage of materials between the nucleus and the cytoplasm. Okay, it is acting as a gate between the nucleus and cytoplasm that controls. Then for nucleolus, it is the center portion. Okay, it is a dense non-membranous mass composed of protein and RNA. Okay, RNA is nothing but ribonucleic acid. Ribonucleic acid, and it produces ribosomals. Okay, RNA for ribosomes. Okay, ribosome which is present normally. Then chromatin. Chromatin is a fibrous strands composed of proteins and RNA. And what is this use? It functions for the genetic code and determines which protein and enzymes will be produced by the cells. Okay, genetic code is, so by this way, by this material we can determine the genetical transformation. Okay, so that's all with this today's class. Hope you understand. We will see the origin of cell potentials in the next class. Thank you.